speaker, Dr. Shakira Hobbs. Dr. Shakira Hobbs is an assistant professor in the Department of Civil Engineering at the University of Kentucky. Dr. Hobbs' scholarship explores multidisciplinary approaches to sustainable engineering, international development, and life cycle thinking applied to the food, energy, water nexus. Her area of research focuses on developing methods to manage anthropogenic activity, such as converting waste to energy and modeling transport of pesticides. In 2018, she founded BioGals, a US nonprofit organization that works internationally empowering women of color to create sustainable solutions. I highly recommend you check it out. Dr. Hobbs received some seed funding from National Science Foundation as well for wastewater assessment for coronavirus in Kentucky, or WACI. So please join me in welcoming Dr. Hobbs.
Thank you, Colleen. It's an honor to be here, and I will briefly talk about working with communities and innovating with food waste and wastewater. The world is healing. We saw environmental conditions improve and more wildlife roaming in cities. Watching this happen during COVID-19 highlighted the stark reality and the effects pollution and waste have on the environment due to human activity. Today, I want to talk to you about two fascinating ways waste can be utilized to help answer COVID-19 related questions and food waste as an energy source. Next slide. Fragments of SARS-CoV-2 found in wastewater can be used to surveillance the currents of people with COVID-19. A group of researchers and I were awarded NIH seed funding to investigate coronavirus in Kentucky via wastewater. The fragments we are interested in are ribonucleic acid, RNA, inside of the virus. The RNA is a living cell, is in all living cells, and its role is to act as a messenger. Next, please. Carrying information about the virus. We can use that information to detect the presence of SARS-CoV-2 in wastewater. Next slide. We collected wastewater samples from the wastewater treatment plant. The wastewater was autoclaved and spiked with synthetic SARS-CoV-2 RNA to simulate a positive person shedding the virus. The samples were put under 3 different environmental conditions. The RNA was extracted from the wastewater using exclusion-based sample preparation and analyzed using polymerase chain reaction. Next slide. 8 different experiments were used to evaluate the aging of SARS-CoV-2 under various conditions. We're looking at these conditions because these are various environmental stressors the samples may be under during transport to the lab for analysis. Next slide. The copies of SARS-CoV-2 are still present at day 3 for most samples. However, no copies are present after a week. This assists with understanding the factors that influence the degradation and time period in which wastewater samples need to be analyzed to make an informed decision. For example, rural areas may be located far from labs for analysis and/or may be in conditions that accelerate the degradation of SARS-CoV-2 RNA. COVID-19 taught us about disparities and how we need effective solutions to help our most vulnerable communities. We've seen this technology be deployed at college dorms, nursing homes, shelters, and prisons. Additional vulnerable communities that could benefit from this are rural communities in low to mid-income countries with septic tanks and trench latrines. Since I've been working with, next slide please. Since I've been working with the rural village in Belize, I became aware of how important it is to involve communities in the decision-making process. I've been working with City River Village in Belize since 2015 and the main method for managing their waste is burning, burying, and dumping trash in the river. The community wanted to explore methods for managing organic waste. Next slide. The spider graph on the right shows current waste management techniques. These are how things are currently ran, and community members want to go from larger area to smaller area. Next. The introduction of biodigesters or anaerobic digestion to manage food waste reduced the area. Anaerobic digestion is a renewable energy technology that converts organic waste, like food waste, to energy. Next slide. With the community, we identify anaerobic digestion as a viable method for managing food waste and utilizing biogas to cook school lunches. We had several interactions with community members, designing the prototype and testing it with the community. We spent a lot of time at the school engaging with primary school kids and introducing them to anaerobic digestion. We built a pilot scale digester with the local construction company. Lastly, women of color that join me on these trips gain a sense of belonging and identify more with being an engineer. And this is just a clip of us loading the digester. Next slide. From sessions 3 and 4, we talked a lot about resource recovery from wastewater. I briefly demonstrated ways we can amplify the value of waste, recover resources, and engage community in technology integration. Due to COVID-19, it's been challenging coordinating international research, but there have been opportunities to support knowledge exchange. Next. Finally, it's important to continue this work because of the impact it continues to have on women of color in engineering. Here are some of the colleagues who accompanied me to Belize and worked on this work with me. All are continuing in engineering as faculty members, postdoctoral scholars, recent graduate, project engineers, and environmental engineer for a government agency. Thank you.